Dear learners, now let us study the physiological benefits of protease inhibitors. Protease inhibitors have been examined extensively as therapeutic agents primarily to deal with various human cancers. Interestingly, many plant-based protease inhibitors are also found to be effective against cardiovascular diseases, osteoporosis, inflammatory diseases, neurological disorders and HIV/AIDS. Let us study them in detail. Now first is protease inhibitors and HIV. Protease inhibitors are used in combination with other antiretroviral agents for the treatment of HIV infection. These drug combinations are synergistic. Protease inhibitors block the activity of the protease enzyme which HIV uses to break up large polyproteins into the smaller pieces required for assembly of new viral particles. While HIV can still replicate in the presence of protease inhibitors, the resulting virions are immature and unable to infect new cells. A study on the beneficial effects of protease inhibitor on body composition and energy expenditure of HIV infected and AIDS patients showed that both patient groups gained weight, body mass index and fat free mass, whereas only AIDS patients gained fat mass. The study concluded that body composition and metabolic parameters improved during protease inhibitors therapy in HIV infected and AIDS patients without wasting. Another study also reported that protease inhibitors therapy of HIV infection is associated with weight gain and improvement in the quality of life indices. The weight gain is mainly in fat mass with no change in lean body mass, that is skeletal muscle. Now, so second is protease inhibitors and cancer. The preclinical, that is in vitro studies and pharmacological effects of plant protease inhibitors in disease prevention have been studied and has been discussed. Begonia seeds are rich in serine and cysteine proteases inhibitors which play an important role in the prevention of common prostate cancer. Viability of breast cancer and prostate cancer cells found to be inhibited significantly by chickpea Bowman-Burke type of protease inhibitor. The purified ragi has a bifunctional inhibitor which reduced cellular proliferation and induced apoptosis of chronic myeloid leukemia cell. The protease inhibitors extracted from common buckwheat seeds induce apoptosis in T acute lymphoblastic leukemia cells with DNA fragmentation. The seeds of soybean possess both BBI, that is Bowman-Burke inhibitors, and KTI, that is Kunitz trypsin inhibitor, which we have studied earlier. Soybean BBI reduced the initiation and regularity of colorectal tumors at low concentrations of 10 mg per 100 gram diet in the dimethylhydrazine rat model without any adverse effect on animal growth or organ physiology. Barley serpins are effective irreversible inhibitors of serine proteases of chymotrypsin family. Trypsin inhibitor protein from sweet potato stimulates the apoptosis of NB4 cells by blocking the cell growth. Lentil seed trypsin inhibitor was able to inhibit the growth of human colon adenocarcinoma and colonic fibroblast cells. The family Moringaceae, that is drumstick, recorded high level of protease inhibitor activity, that is about 92% against trypsin. The proliferation of HT29 colon cancer cells was notably affected by protease inhibitors from peas in a dose dependent manner. The protease inhibitor extracted from potatoes, that is POT2, increases cholecystokinin, that is CCK, release on food intake. The endogenous CCK may be important in the control of food intake, and that protease inhibition may have therapeutic potential for reducing food intake. A trypsin or chymotrypsin inhibitor from seeds of Vicia faba, that is field bean, showed ability as effective chemoprotectors against gastric cancers in animals and possibly in humans as well. Skin carcinogenesis can be effectively suppressed by topical treatment with a FBPI. The black-eyed pea chymotrypsin inhibitor, that is BTCI, from cow pea seeds induces apoptosis, that is cell death, due to the morphological alterations of the cell and lysosome membrane permeation demonstrated via cytostatic and cytotoxic findings. Numerous protease inhibitors have been isolated and characterized from different plant species like peanut, pigeon pea, black gram, mustard, maize, cucumber, watermelon, spaghetti squash, red bryon, fig leaf, wild cucumber, wax gourd, rice, maize and apple fruit. Now third physiological effect is protease inhibitors and gastrointestinal diseases. The therapeutic potential of protease inhibition in gastrointestinal diseases with a particular focus on inflammatory bowel diseases, functional gastrointestinal disorders and colorectal cancers have also been found useful. Now let us see the adverse effects. Adverse effects associated with the use of protease inhibitors are nausea, vomiting and diarrhea. Metabolic side effects include glucose intolerance, diabetes, hypercholesterolemia and hypertriglyceridemia. Prolonged use has been associated with fat redistribution. Now let us also study protease inhibitors from legumes. Protease inhibitors from legumes can interfere with protein digestion, leading to growth retardation, and in some species of animals can cause pancreatic enlargement and enhance chemically induced pancreatic tumors. As we have studied earlier that two types of trypsin and chymotrypsin inhibitors have been identified in soybean, that is Kunitz trypsin inhibitor and Bowman-Burke inhibitors. Boiling dry beans generally reduces the protease inhibitor content by 80 to 90%, and there is little reason to think that the amount of trypsin inhibitors obtained by eating commonly consumed beans would exert any adverse effects in humans. Now contraindications. The protease inhibitors are contraindicated in those known to be hypersensitive to them and should be used cautiously in patients with liver disease, hemophilia or diabetes. Now the last part is perspective of uses of protease inhibitors. Protease inhibitor is the potential source for the development of a new drug against thrombin, elastase, chymotrypsin, trypsin, cathepsin and pepin in pharmaceutical industries. Furthermore, these protease inhibitors could also be used as a seafood preservatives against proteolysis and in other biotechnological applications. Now let us conclude. Many protease inhibitors have been found in natural sources and also have been synthesized for clinical uses. Among all, soybean Bowman-Burke inhibitors represent the most extensively studied but related BBI from other legumes including chickpea, common bean, lentil and pea, and from wheat, rice, barley has been well recognized and characterized. There are several advantages of using plant protease inhibitors compared to synthetic ones. Plant protease inhibitors can also be supplied through diet, for example through rice, potato, legumes, soybean, corn, cucurbits, cereals, etc. by adding some extra plant based food preparations which will have no side effects on human body. Extensive research has to be done to find out the possible candidates of protease inhibitors that have therapeutic importance from plants. With the growing population and diseases, there is a need to explore more plant protease inhibitors useful in the treatment and control of human diseases. Thank you.